Now we will talk about the third component of a generalized cell, which is the nucleus. Nucleus is considered to be the most important part of the cell because it is like the brain of our body. Just like the brain controls the body, the nucleus controls the cell. It regulates and coordinates various life processes of the cell. It plays an important part in cell division, just like the brain plays an important part in the reproduction, and it contains the genes or the factors which determine heredity. It basically means that nucleus is a small spherical mass which is located somewhat in the center of the cytoplasm. It has a very delicate nuclear membrane and it is filled with a nucleoplasm. If you recall, nucleoplasm is non-living in nature. Besides that, it is like cytoplasm but relatively denser. Within the nucleoplasm are certain thread-like structures which are called chromatin fibers. When cell division takes place, the chromatin fibers become thick and ribbon-like, and then they are called chromosomes. Now, there are certain cells in which the nuclear membrane is absent, and such cells are called prokaryotic cells. Pro means primitive and karyon means nucleus. That means it has a primitive nucleus. The chromatin fibers in such cells are dispersed freely in the cytoplasm. An example would be bacteria. The cells in which double nuclear membrane is present are called eukaryotic cells. All organisms other than the kingdom of bacteria or Monera have eukaryotic cells. If you notice figure 2.8, you will note that there is a cell in which the nucleus has been highlighted. The centrioles are moving apart in order to divide the cell. The cytoplasm has been indicated and the chromosomes have been formed from the chromatin material. This shows how a cell looks like when division is about to take place. A nucleus also contains at least one nucleolus. Now, nucleolus is like the brain of nucleus. Some cells may also have more than one nucleolus, but the number of nucleoli in a cell is fixed. That is, if one nucleolus is present in the nucleus of one cell, the entire body will have the same number of nucleolus. It participates in protein synthesis. The number of chromosomes also are fixed in each species. So all human beings have 23 pairs or 46 chromosomes. Following is the number of chromosomes which are present in a variety of animals and plants. It is surprising to know that some insects which are much smaller than human beings have more than 1,000 chromosomes. The chromosomes have a very important role to fulfill because they carry the genetic characters from the plants. The chromosomes have a very important role to fulfill because they carry the genetic characters from the parents to the offsprings through the fusion of the egg of the female and the sperm of the male. Chromosomes are composed of chromatin, which is made up of genes. So basically, genes make up chromatin material, which make up the chromosomes. Genes are made in turn of chemical substance known as DNA, and we all know that DNA is the full form of deoxyribonucleic acid. So the DNA makes up the genes, which makes up the chromatin, which makes up the chromosomes. You can further read about DNA fingerprinting and its role in forensic sciences in today's time. You also need to remember that genes and not the number of chromosomes determine the characteristics of a species. Take for instance dogs. Dogs have 78 chromosomes and so do wolves. but their characteristics are different even though they are from the same species. That is because they have different types of genes and different types of characteristics. However, the number of chromosomes in them are same. You can also take the example of lion, tiger, and the house cat. In the next part, we will study about the similarities and differences in the plant and animal cells. By now we know that both of these contain some organelles which are in common, and there are some organelles which are different. They both contain cell membrane, cytoplasm, nucleus, endoplasmic reticulum, Golgi bodies, mitochondria, and ribosomes. And when it comes to differences, the differences in them are in cell wall, centrosome, vacuoles, plastids. And when it comes to differences, they have differences in terms of structure and in terms of quantity. In terms of structure, there is a difference of cell wall, centrosome, vacuoles, and plastids. Plant cells have a cell wall while animal cells don't. Similarly for vacuoles and plastids, which are found in plant cells but not in animal cells. Vacuoles may be found in animal cells but they are small and temporary. On the other hand, centrome is not present in plant cells but it is found in animal cells. When it comes to quantitative differences, the plant cells are usually larger with distinct outlines, while it is not the case for animal cells, which are usually smaller and with less distinct boundaries. The cytoplasm is not so dense in plant cells, but it is denser and more granular in animal cells. When it comes to arrangement of cytoplasm, as we talked about before, only a thin lining of cytoplasm, which is found mostly pushed to the periphery, is found in plant cells. And why is that so? If you recall, that is because vacuoles are very prominent in plant cells and quite large in number, because of which the cytoplasm is pushed to the periphery. On the other hand, cytoplasm fills almost the entire cell because of the absence of vacuoles in animal cells. Next, we talk about protoplasm. Putting it simply, protoplasm contains all the things of a cell other than the cell membrane and the cell wall. Protoplasm is equal to the cytoplasm plus the nucleus. Protoplasm is basically the living substances in an organism. Now, cell membrane is also living, but it is not considered to be a part of the protoplasm. The cell composition of protoplasm is very complex and it varies little from one cell to another. And the common elements included are carbon, hydrogen, oxygen, nitrogen, sulfur, iron, phosphorus, and they are all same in all cells. There are also some specific compounds such as water, proteins, carbohydrates, fats, and mineral salts. It is also important to know that it is impossible to make an accurate chemical analysis of protoplasm because as soon as it is removed from the organism, it stops being protoplasm. Again, there comes the definition of a cell. We have already talked about it, and we can also talk about the differences in eukaryotic and prokaryotic cells. We can note that here is the final definition of a cell. It is the basic structural building block of all living organisms consisting of protoplasm enclosed by a cell membrane with an additional cell wall in plant cell and having a nuclear membrane or without it. We can note the differences in the highly diagrammatic sketches of eukaryotic cells and prokaryotic cells in figure 2.9. Also is given the difference in table 2.3 and you can go through it. They are quite simple. Also is given the difference between prokaryotic and eukaryotic cells. In prokaryotic cells, there is a nucleoid, which is basically a nucleus which is not very well defined, while in eukaryotes, there is a well defined nucleus along with a nuclear membrane. And also in prokaryotic cells, a single length of only DNA is found, while there are several lengths of genetic material containing DNA wound around certain proteins. So it would be something like proteins, and there is DNA wound around it. Then there is some more DNA, and they are bound via protein. Then like this again DNA bound around a protein, and so on and so forth. And this is how the DNA, when it's not in the cell division phase, looks like in a eukaryotic cell. The ribosomes are smaller in prokaryotic cells as compared to eukaryotic cells. Also in prokaryotic cells, no other cell organelles are found, while it is not the case in eukaryotic cells, in which several organelles like mitochondria, endoplasmic reticulum, chloroplast, and all the ones we have read before are present. The examples of prokaryotic cells are bacteria and blue-green algae, and all the other organisms in the living world contain eukaryotic cells. You can notice that the spelling here is not correct. It should be EUKARYOTIC. Please correct it in your books. Now is given the activity outcome of cellular activity. You can read about it. It is quite simple. So the growth of organisms, the repair of injury, movement of the body, be it walking, running, jumping, flying, the feathers used in flying, drooping of leaves, bending movement of roots, closing of stomatal leaves, feeding and nutrition, including swallowing, tasting, digestion, absorption, circulation of blood in the muscle cells of the heart and other parts, respiratory gases, the transportation of it, the protection of body against germs and bacteria, the sensations, the thermoregulation, reproduction, absorption of water and nutrients through root cells, the trapping of light by leaf cells, the attraction of insects by the flowers with the help of their colors, and the transmission of parental features from parents to their young ones. They are all as a product of cellular activity. And while all the living organisms perform activities with the help of cells, they are performed by, and those cells are specialized for particular functions. Salient points of this chapter. First off, we studied what a cell is. A cell is the fundamental structural and functional unit of all living beings, and it is the building block of all living organisms. Next, we studied about the invention of the microscope and hence the discovery of cell. In this, we talked about Antony von Leeuwenhoek, Robert Hooke, the compound microscope given by Hooke and today's compound microscope, and the electron microscope. Next, we studied about cell theory given by Matthias Schleiden and Theodor Schwann, and the addition of it by Rudolf Virchow. We studied about the cell theory, in which it is said that the cell is the smallest unit of structure of all living things. The cell is the unit of function of all living things, and all cells arise from pre-existing cells. Next, we talked about the quantity, the size, and the shapes of cells. The most important part of this chapter includes the structure of a cell, in which we talked about a generalized cell which consists of cell membrane, nucleus, and cytoplasm. We also talked about cell organelles, which are like the organs in a human body. We then came down to the parts of a cell, in which we differentiated between living parts and non-living parts. And we talked about which of these are present in both the animal and plant cells and which are present in either. We can revise each of these parts with the help of table 2.1. First off is the plasma membrane. It is also called the cell membrane. It is the outermost layer in animal cells, and it lies next to cell wall in plant cells. It is very thin, flexible, and a living membrane. It possesses fine pores and it is semi-permeable in nature, which means it allows some substances to pass through it and does not allow the others to pass through it. And it is also important to remember that it is made up of lipoproteins. Its chief function is to separate contents of cell from its surroundings. It regulates the... We will now come down to the most important part of this chapter, which includes the parts of cells, their main functions and chief functions. We'll now come down to the most important part of this chapter, which is given in table 2.1, which includes the part of cells, their main characteristics and chief functions. First off is the plasma membrane. It is found both in animal and plant cells, and it is the outermost layer in animal cells, but it lies next to cell wall in plant cells. It is semi-permeable in nature and has fine pores. It is living in nature. It regulates the entry of certain solutes and ions, and it maintains the shape of cells. Next is the cell wall. It is found only in plant cells. It is mainly composed of cellulose and it is non-living in nature. Other than that, it allows the entry of all substances through it. Its main function is to provide rigidity and shape to the cell. Next we study about cytoplasm. And cytoplasm is the semi-liquid fluid in which all the cell organelles float in a cell. It is found in greater quantity in animal cells as compared to plant cells, and in plant cells it is pushed to the periphery due to the presence of vacuoles. Its importance is that all metabolic activities occur in it. Other than that, next we have the endoplasmic reticulum. Endoplasmic reticulum, also known as ER, is an irregular network of tubular double-membraned structures. It is connected both with the cytoplasm, plasma membrane as well as the nuclear membrane. It may be smooth or rough in appearance based on whether ribosomes are attached to it or not. Its two main functions are that it provides supportive framework for the cell and it facilitates the synthesis and transport of proteins and fats. Next we have the mitochondria. It has various shapes, but it is usually sausage-like. It is double-walled, it has its own DNA, and it also contains its own ribosomes. Its main function is to release energy, and it is the seat of cellular or aerobic respiration. Next we have the Golgi Apparatus. These contain stacks of flattened membrane sacs, and they may contain tubules, vesicles, and vacuoles. They facilitate the synthesis and secretion of enzymes, hormones, etc., and they form acrosome of sperm. Next we have the ribosomes. They are small granules which are either distributed in the cytoplasm, that is, they are found freely floating in the cytoplasm, or they may be found attached to the outer surface of ER, that is the endoplasmic reticulum. They are also single-walled, dense, spherical structures which are composed mainly of RNA. To reiterate, RNA is ribonucleic acid. Its main function is protein synthesis. Next we have lysosomes. These are the membranous sacs which are budded off of Golgi bodies. So lysosomes are basically a part of Golgi bodies which have been separated from it. And they contain 40 different types of enzymes because they are imperative in intracellular digestion. They also destroy foreign substances, and they can even destroy organelles, and hence they are known as suicide bags. You can compare lysosomes with the stomach of human beings. Next we have the centrosome. It is to be noted that they are found only in animal cells and not in plant cells. It is a region surrounding the centrioles located near nucleus. They contain one or two centrioles, and centrioles are surrounded by radiating microtubules to form a star or an aster during cell division. They are mainly responsible for initiating and regulating cell division, and they form spindle fibers with the help of asters. Next we have the plastids. Again, plastids are found only in plant cells and not in animal cells. They contain three types of plastids can be of three types based on the pigment they contain. So they can be leucoplast, chromoplast, and chloroplast. Each of these have their own function, such as chromoplast imparting color to flowers and fruits, chloroplasts trapping solar energy, and leucoplast for the purpose of storing starch. Next we have the nucleus, which is the largest cell organelle and also the most important one. It is spherical and dense in nature, and also it has pores to allow substances to enter and leave it. It is very important as it regulates cell function. It contains the chromosomes, and if removed, the cell dies. It is very important as it not only regulates the cell functions but is also responsible for controlling the hereditary characters contained in the chromosomes. Next we study about nucleolus, which are one or more round-shaped nucleoli inside the nucleus. Their main function is to produce ribosomes. Participating in protein synthesis and dictating ribosomes to synthesize proteins. Next we talk about chromatin fibers. Chromatin fibers are made up of DNA threads, and they are the network in resting stage of nucleus. Next we talk about chromatin fibers. They are basically the network in resting stage of the nucleus, and they condense into chromosomes during cell division. They are made up of DNA threads, and chromatin fibers are very important as chromosomes carry hereditary information or the genes. While all the parts we study till now are living in nature except for cell wall, the next two that we talk about are non-living, but they are also very important. Vacuoles are clear spaces with water or other substances within the cell. Plant cells contain large vacuoles, and the animal cells have fewer and smaller ones, and they may be temporary. It is important to remember that they are covered by a covering called tonoplast. Its functions include the storage of water and giving turgidity or rigidity to the cells. Next we talk about the granules, which are small particles, crystals or droplets. They are considered to serve as food for the cells. We also need to remember the differences between plant and animal cells. First are the structural differences in the form of cell wall, centrosome, vacuoles and plastids. And the other difference is in terms of quantity, which includes the size, the cytoplasm, and the arrangement of cytoplasm in the cell. The difference between prokaryotic and eukaryotic cell, and the outcome of cellular activity in living organisms. The next chapter will talk about various kinds of tissues in animal and plant bodies.